What is going on, everybody? Down to Funk here. Coming back at you with another DtF and Co's Ohio Grow Show. Hope everybody can hear me pretty well here. We got the coffee. We got the Dabarooskies. We got... You know what? I'm not even gonna lie to you. I got a mic stand now, and it's technically my bong, so got him. Tobacco bong, of course, but that's neither here nor there. Let's see. We on? Brewskis, we got. Oh shoot! Sorry about that, guys. I want to hop in the live chat. Botrix, what is going on? Well, first and foremost, everybody, let's load something up right quick here. So. What's good, Anthony? What's good? How's it going, everybody? Certified. Oh yeah. How's it sound to everybody? Does it sound okay? Trying to make sure I get this dialed in this time. What's everybody smoking today? Get this dab packed up real quick. Oh yeah, let's get it. Let's fucking go, dude. Blue Dream, nice. One of my homies said he's about to hook me up with some Blue Dream auto flowers, and I was like, oh shit, dude, hell yeah. I'm about to smoke on some Golden Gauge dabs. They're pretty damn good. I like them. Really tasty. It's really convenient just to be able to load it up real quick and hit it real fast on the little e-rig while I'm doing stuff like this. So that's what we're doing. Purple cheese cookies and jelly roll. Nice, bro. That sounds fire. Mystery dabs are always cool. I like mixing them sometimes, putting them together. It tastes pretty good. But yeah, let's do a couple of hits real quick before we get started. You already know what time it is. Hope everybody's having a great weekend. I hope everybody's staying warm. Big chillin'. Hope everybody had a good Valentine's Day and stuff if you celebrated it. And if you didn't, I still hope it was good. OG Kush. Nice, dude. Hell yeah. Of course. Yeah, of course. I already knew that, of course. Gotta love the homegrown, son. Actually, speaking of homegrown, just got some of mine chopped down last night. So that was pretty fucking exciting. So, gonna chop down some of the Georgia pies today, the rest of those, because they were still looking like they wanted to go another day. So, gonna get that done tonight. Thinking about doing a live little chop down, but we'll see. It'll be kind of hard to record it and do it, but we'll see. I'll try to do it. Oh, happy birthday, Bluesman. Happy birthday. Everybody say happy birthday to Bluesman. Nice, brother. Get it done. Get it done. I'm about to get mine done, too. Happy birthday, brother. That's what's up. Thank you, Anthony. I appreciate it. I'll send some. I think I sent some of the pictures to the Discord, but I'll make sure I send some pictures and make a little video compilation or something. Definitely doing a smoke report. I'm re-vegging the One Milktopia that I really wanted to get and keep. So I'm doing that one, and then I'm keeping one of the Georgia pies too. I just haven't really made up my mind yet of which one, so gotta see on that. Nice, dude. Hell yeah. Hey, happy birthday, George. Happy birthday, brother. And Bluesman, yeah, that was the Coltogen Seed Co. Regular auto flower, yeah. And yeah, it was the broomstick auto. Alright, you guys, I'm loading up the dab right now. If you want to hit one real quick with me before we get started. She's loading up. T minus like 10 seconds probably. Cheers. Oh yeah. Oh shit. Dropping stuff, guys, sorry. Cashew notes, pine and floral, incense kind of blend. Nice. That's what you want right there. That sounds great. Woo. Some good hits right there. Yeah, I appreciate you, Grown. I appreciate you, brother. Coltogen hooked us up. That's really what it was. Quite honestly, he hooked us up real good. But yeah, no, I agree. I agree, dude. Should be really, really milky, I'd imagine, George. Oh shit, that hit got me. Yeah, we're about to get started, guys. Here, just wanted to chop it up with you guys, see how your weekend was, check in with you, make sure that everything's going good with you. Make sure there's no hiccups, and you know, everybody's just big chilling. So glad to hear that. Oh, I also want to give a shout out to my homie JoJo. He's a motherfucking man. He hooked me up with these little dope background pictures and themes and stuff. So gonna start trying to implement a little bit more color and flair into the podcast just to make it a bit more professional looking and stuff. So yeah, hope you like it, and shout out to JoJo. No, you're gonna find something in there, dude. I'm sure you will. I'm sure you will. I'm trying to get Coltogen to get up on the Discord. He said he normally doesn't really mess with it, so trying to get him to get on there so he can really see everybody's different phenos that get popped and everything, because I think that'd be a really cool thing to have him in there, and he'd be like, oh yeah, I saw this or I saw that when I was, you know, doing my stuff. So that, trying to get him in there, but he's a busy guy, so. Alright, I think this dab probably got maybe one more load up. I'm gonna reheat this, then we're getting started, you guys. Don't want to take up too much time of your day. I appreciate everybody being here, each and every one of you. T minus like 10 seconds. They'll be ready. That sounds fire, Bluesman. Hell yeah. Hell yeah. Alright, cheers, everybody. Oh shit. All right, let's get this show started, son. Welcome to the DtF and Co's Ohio Grow Show, episode number 3. So today we're going to be talking about Mendelian genetics, presented by yours truly, DtF, down to funk. This is my favorite picture. I don't know if you guys noticed the playlist, the podcast playlist, but this is by far my favorite picture that JoJo made for sure. He definitely hooked us up, and it's definitely a good one right here. So shout out to him once again. So yeah. Once again, I want to give a special thanks to people that support me, because you gotta support the people that support you. So I want to give a shout out to Cultigen, Seco, P&W Cultivar, especially just those two for believing in the channel, hooking us up. You know, they're the ones that went ahead and got the seed packs sent over to us so we could do some giveaways and all that. So, you know, shout out to them, and I have all their information tagged inside of the bio, so make sure you hit them up and see them, and Dab Marino as well. His packs are already given out and those are gone, so shout out to him always though. Thank you Cultigen for the knowledge and for your support. Thank you P&W Cultivar for your knowledge and your support. And shout out to Zaza Genetics today. So I want to give a shout out to Zaza because he actually inspired the topic of this episode. So today I basically wanted to break down what genetics is and what Mendelian genetics is, because Gregor Mendel is the father of genetics, is what they say. It's just what they teach in classes, and he had a big part of it. So the reason Zaza inspired this is because he had a video come out not too long ago that was called Breeding with Intention, Making the First Selection Your Best. And if you look at my bio after this is over, go ahead and go check that video out for sure. And in there he mentioned Mendelian genetics, and he mentioned the stuff we're going to talk about today. So shout out to him, and yeah, and thank you to everybody that gave encouraging words, and thank you to everybody that supports the channel, and you guys make this happen, so I appreciate y'all. Make sure you go show love to everybody, for sure. And yeah, all the links, like I said, are in their socials, or in the description. Always got coffee going. Always got coffee. So, brief summary of that video. So he was speaking about Mendelian genetics, like I said, and we're going to talk about that today and how that ties into the Beginners Breeders Project, and why it's important to building a strong foundation of basic plant breeding before fully jumping into the world of breeding. You know, it's cool. Like, I accidentally had my first 2 crosses, and that's what spawned my interest and encouragement to start doing this type of stuff. So I just want to make sure if I'm doing it that it is done in the correct manner, and I want to spread good knowledge. So we got to start from the beginning. It kind of sucks sometimes, but you got to go through the process to learn, and you have to always be willing to learn, because there's always new stuff to learn. So it's the process that's the hardest. So as long as we can break it down so we're not confused, and you know, and we're including everybody in the conversation, and we're making sure we're just aware of what's going on, then it should be pretty cool conversation we're gonna have today. And yeah, so I'm gonna turn off my dehumidifier. Hold on. Alright, I'm back. So, my goal is to provide information and perspectives to build on each week from the last podcast. And I would like to add on, like I said, and just constantly give updates on the grow and then on what we're building from last time. So, yeah, thanks for being here. So, introduction here. Who is Gregor Mendel? So, a little biography on him. He was born in 1822. He was an Austrian monk / botanist. And he died in about 1884. And he had a really good education. He started out just going to school, and he was very academically successful. And then he went on to go to the Philosophical Institute of Olmütz, and then he shortly became a monk not too long after that. And then they eventually sent him to the University of Vienna, and he finished his education there, and then he went back there, and from around 1853 to 1863 is when he was conducting the first kind of really recorded for what we're going to be talking about, which is Gregor Mendel, the first recorded experiments on genetic plant breeding. He was breeding pea plants, and he chose pea plants because of their short reproductive cycle and the many varieties, and he was very interested in what he could do with these plants. So he conducted his experiments in his free time here at this monastery, and he had really good notes, and he kept really good notes, and he had a lot of great knowledge and implemented that knowledge, and here we are today. So that's life. That's full circle right there. I try to reference everything in the like slide page, and then I have a reference page at the back too, and then I'm gonna go back after the stream is over and put all the references, the links, if everybody wants to go read some more of this stuff. I'll put all the links inside of the YouTube page, and then you guys can check out that stuff too if you want to read a little bit more and everything. But yeah, that's a little basic synopsis on it, just a brief knowledge on them. So yeah. So Mendelian genetics, what is it? Well, for our purpose, we need to think about what's genetic breeding. So he was breeding these pea plants, right? So genetic breeding is plant breeding because you're crossing two things that have genes in them, and then for Mendel's purposes, he was crossing them again, so he was genetically breeding. So genetic breeding, also referred to as selective breeding, is taking desirable traits from two parents with the intent to produce more desirable traits in its offspring. And the traits that are passed on are called alleles. So traits and alleles are interchangeable. It's the same thing. So it's going to be interchanged and said quite numerously through here. So just remember, trait and allele is the same thing. So there's dominant alleles, which that determines the trait that's expressed. So if you have a dominant trait in comparison to a recessive allele, which is a trait that is suppressed by the dominant allele, that recessive allele is not seen. It's still passed on in the offspring of the plants. So when you take two plants and they have seeds, there's still, you get some stuff from the mom and you get stuff from the dad, but there's still stuff that if you went back and crossed those seeds again and you get more seeds from them, they're gonna have more traits that come out that maybe you didn't see the first time. So that's what a recessive allele would be. So that's just a basic thing on that. So take a drink of this coffee here. So this type of experiments that he was doing, it led him to developing Mendelian inheritance, which is basically just certain patterns of how traits pass from parents to offspring. So the traits would be the alleles we just discussed, and he was concerned with why that was happening. He wanted to know why that would occur whenever they would be crossed together. So that's what we're gonna try to break down, and then you can just put that in the same context as plant breeding. So it doesn't matter if it's a pea plant or corn or cannabis. It doesn't matter. It all applies because it's genetics. So sorry about that. Keep coughing. Hit too many dabs. So in this, as he was doing this, he developed 3 laws of inheritance by 2 different experiments. He did this thing called a mono hybrid cross, and then he did a di-hybrid cross, which we'll get into more of that about that. So before that, let's discuss genotype and phenotype to get a better understanding of the terminology and how this is a part of Mendelian genetics. So just like the variations in the pea plants can be seen individually in each seed, or cannabis seeds, you can see different variations from the same seed, plant the same seed, and then you look at them side by side when they're growing, and 2 of them can be completely different. But they came, for sure they're from exactly the same thing. So that's kind of what this is going to be about. But you see different phenotypes or genotypes being shown because there's different dominant alleles and recessive alleles being combined whenever those seeds were initially formed and made, and in that process, that's how that seed just has encoded in its genetics that when it grows, it's going to express this way, and then even though its brother or sister, if it's a regular seed and not a feminized seed, if its brother or sister has a different genetic code, then it's going to express a different gene expression. So that's why it has a different look or phenotype. So we'll get into all that, but that's just a little summary of what we're going to be getting into. So genotype versus phenotype. What is this? Well, a genotype is the genes that an individual carries and passes on. So the genes that make a plant tall or short. If you want to, you can think about it in the pure 100% landrace sativa or the 100% landrace indica. How they say that the sativa ones grow a lot taller and lankier with a lot narrow and more pointy leaves, and then the indica have more broad leaves with short, compact growth. They have very short stature. So that's the genotype. These are all observable traits, of course, when you look at the plants, but they're determined by the genes that come from when that plant was formed, whether or not it had a dominant allele or recessive allele combination, and whether or not that's now being expressed. And that's why you can see if it's a narrow leaf or a broad leaf. But yeah, so these are all observable traits, which the traits are the phenotypes that you can see. That's the observable trait, that's the phenotype. And this is determined by the genes that the landrace is carrying, so that's the genotype. So that's why it's expressing those physical attributes. So phenotype, like we just explained, is all observable characteristics, but this can be driven by your environment as well as the genes that the plant has. So this explains that the physical attributes, the phenotype of the plant, is directly influenced by two things: what the genes or the genotype of the plant is carrying and now expressing, and that's what you're seeing through the physical phenotype or observable characteristics, or and yeah, I should say or slash and the environment the genes are expressed in will determine which genes are expressed. Oh shoot, sorry guys, let me go back. Express the plant. I just realized I got a typo in there. That are expressed in the plant. So think about when a plant becomes purple or has different colors instead of like green. This is something called anthocyanins, and it's in a lot of different plants. It's like why strawberries are red, or like why, you know, like I just said, why cannabis is purple, or sometimes light green or dark green. And this is in response to stress, you know, and sometimes the stress will cause it to turn purple, and that's caused by the anthocyanins, and there's over 600 different anthocyanins, and through these different anthocyanins, you'll have different physical attributes that are coinciding or being expressed with that anthocyanin, because it's that's what it does. So here we got some information about this here. So let's think about the sativa and the indica for a second, just because that is at the end of the day what we're talking about. But Mendel is just a really good starting point, because that's the beginning. So the sativa plant originated and adapted to warm environments and climates that it came from, and the reason that they have short, narrow leaves is because the plant doesn't need to get as much sun, with it being that if it was adapted and evolved in a environment where it's sunnier in an equatorial place, like in the southern hemisphere, when it's warmer a lot longer than, let's say, the Midwest here, like when I'm in where I live in Ohio. If it's a lot warmer there all year round, then the plants that are acclimated to live in that climate are gonna be different here. That's why you can't take a plant from out there and then bring it out here and expect it to grow all year round. So unless you have some type of greenhouse or something outdoors that you're doing and all that. But so the reason that the leaves are so narrow is because it doesn't need as much surface area to be able to do the process of photosynthesis, which is the way the plant uses the sunlight and then converts that into energy. So that's why the leaves are a lot more narrow. It doesn't need to take up as much sun because the sun's there all the time. So the plant knows that, so it's adapted over time to not have to direct energy into growing these big broad leaves, as you would see in the indica plants, which is the opposite of the sativa. So if we look at the indica, 100% pure indica sativa or indica plants, like we discussed, they have really broad leaves that are short, short in growth and really compact. And a really great example of that is if you go on my YouTube or my Instagram, is the apes in space, which is bred by Exotic Genetics. Its lineage is grease monkey, and I believe, I want to say lemon tree, but I might be wrong, guys, so don't quote me. But I know grease monkey for sure, and I believe the grease monkey is what gives it its really short stature. And if you compare that plant to the rest of my other plants, you can really tell that it's not just because the buds on it were really beautiful purple, and they seem to be, we're going to see after dry if they're going to still have this look, but they seem to be like they're pretty okay size. Once again, I know that what happened at the beginning of my flower was that I didn't push extra like my regular compost teas that I would do for my vegetative cycle, and I went straight to my full flower cycle compost teas, and I could have just kept doing the regular vegetative one with the stretch that occurs naturally with the first 1 to 3 weeks as you switch to flower. So I kind of lacked on yield because of that, and maybe not as many bud sites or as big as of bud sites as I wanted, but it's awesome for me. I'm really excited. So I think one of the best things about growing is that as long as you're excited and you're satisfied, that's one of the best parts about it. So and I'm satisfied for now. Well, I guess it's too early to tell because I gotta finish drying it and curing it first, and then we'll really see. So but yeah, so let's get back into this. So the indica, they adapted over time in harsh environments with a lot colder temperatures. So the reason that they have broader leaves is because that the leaves needed a bigger surface area to take up more energy, or to take up more sunlight, and to go through photosynthesis to make more energy for the plant. So it wouldn't make sense for them to if what produces energy for them is to have the sunlight hit its leaves, and then the leaves then convert that into energy. It doesn't make sense for it to have really narrow leaves like a sativa plant. So that's why the leaves would be a lot broader. So indica plants are just like you would see a variation, like we've talked about, in different cultivars of the same seed pack. If you plant one, and one might be a little bit bushier, one might be a little bit better at have a better structure, or one's a little bit more lackluster on the bud form. Like I have a Georgia Pie one that is the buds are really cool like colors, but they're like, I can tell they're like a little bit larvae in comparison to the other ones. So think about it like this: if you get an indica plant that has a lot of narrow leaves and that's super super tall, it's not going to survive in a place like the Hindu mountains, like up there. So that's why it over time, those type of seeds that would be grown, if they would make it for so long, they would eventually just die off, because those other plants would be more efficient at producing energy and surviving, essentially. So here's a really cool fact about the anthocyanins. So it's a group of deep red, purple, and blue pigments found in plants. They're part of a larger category of plant-based chemicals called flavonoids, which some of us might have heard about that. And that's a different subject. We're going to stick to what we're talking about, but it's just a cool little fact. Flavonoids are abundant in all parts of plants, so fruits, seeds, shoots, flowers, and leaves. They help plants reproduce by attracting pollinators and protect plants from environmental stressors like ultraviolet light, drought, and cold. They also can contain high amounts of antioxidants. There's 6 major anthocyanins, and that would be cyanidin. And according to Bossolino, in all 2023, cyanidin. Yeah, let's just say that. 3 rutein inside seed side is the major anthocyanin within cannabis. And then we have delphinin. Oh man, I might took too many dabs. We got delphinidium, and then we got malvidin, and then perloginidin, and then peonidin, and putunidin. Bear with me. So next slide. Mendelian genetics continued. So in Gregor Mendel's experiments, and the reason the anthocyanins are important in this is because he would be taking different plants of his pea plants that, like, let's say one was tall or short, or one was green and yellow, and then he wanted to know if he could take a tall one and then cross it with a short one and then make it and then they would have short offspring, or if he could have a short one have tall offspring. So as he was doing this, began to notice that there was stuff going on, and there was repetitions of his data that was occurring. So he double backed and he started to call the first generation that would produce if he crossed a tall plant with a short plant. He called that first generation the F1 progeny. And then when this happened, he had all tall plants, and then he crossed the F1 progeny, and then there was tall and short. And then in this F1 progeny, there's a 3-to-1 ratio of tall to short, and then he just kept doing these crosses because he wanted to keep going with seeing if he could figure out why this was happening. So that's the monohybrids. And the dihybrids, the two traits that are considered is the dihybrid cross. So the mono would be one trait. You only have one trait. He wanted to take the tall and the short. He was only concerned with that. And then a dihybrid would be if he wanted to do a wrinkled green seed cross to like a smooth round yellow seed. And he was trying to just see what would happen. So the F1 progeny changed the colors of the pea pods to yellow round. And he didn't know why this was going on, and once again he kept repeating this, and he kept recording the data. So that's something that you might have heard people talk about before. It's very important to take notes and to keep a lot of good data, because that's going to end up helping you if you're growing the same stuff, or even if you're growing different stuff. You can compare your notes on how different cultivars grew and did in different environments during different stages of growth. So that's another thing that was also going on. So as this went on, the dominant traits were then the round shape and the yellow color, and then from this he self-pollinated the first generation of offspring. And then there was 4 different traits because of the dihybrid cross. There was round yellow, there was round green, there was wrinkled yellow, and wrinkled green pea pods. And then the ratio to that was 9 to 3 to 3 to 1. He took very extensive notes, and he had a lot of time to do these experiments, and that is the basis of what is Mendelian genetics. And from this he continued onward here, and he started to make more conclusions of it. This is just a diagram, and this is all the information is linked to what this is, and where this is referenced from at the end, and you can see in the corner right there where I got this information from. But this is just to show different ones of the pea pods that were going on here. So you guys can just take a second to look at this here. Give it a few more, about another minute here. We'll leave this up here. We got 4 different categories. We got seed, flower, pod, and stem. Okay, so this is what Gregor Mendel is working with. So in this next one here, if you look at figure A, this right here is what he was calling a true breeding line. Which he was crossing two of the same. So it'd be like this, crossing two blue dreams to each other, a male and a female. And then he kept doing that, and that was a true breeding line. And then in diagram B, this is a monohybrid cross, which is produced by mating two different pure breeding lines. Reference below. So as you can see, there's a purple flower crossed to a white flower. And then in the first generation of offspring there, the F1 progeny, there was mainly purple flowers that came from the pea pods. If you look at the true breeding line, if you look at those all capital A's, and then you compare them to diagram B, where there's a capital A's and then capital A's for the purple and then lowercase a's for the white ones. That's because the white ones would be the recessive trait, and then the purple would be dominant. And then that's why in the first generation of offspring here you would get purple, but then in the next generation, if you crossed them again, you would then get purple and white. So one reason that he chose the pea pods is because it has a really good and short reproductive time. And that's just a little small diagram here. So just kind of something to break it down a little bit. But the X just represents the two capital A's and the other two pair of capital A's cross into each other, and then since those are both in all capital A's, the only thing being expressed in the true breeding line is mainly just the purple pea pods. And then same with let's see over here on the monohybrid side, since there's one trait that's different. We have the purple flower and then the white flower, and then you cross those two together, and then you get still mainly, because the dominant trait is the purple flower again and the recessive one is the white flower, you still get purple, but then you cross them again, and since that recessive trait is carried on still in the background of the purple flowers, even though they're still purple. Now, and you can think about it in the aspect of, like, if you have a sibling, or your parents have siblings, they don't look this exact, unless they're a twin, an identical twin. They don't look exactly the same, but they still have similars that are all resembling, and you can tell, oh, okay, yeah. So that's kind of a way to look at it too. Let's go to the next diagram, or next part. Yeah, next diagram. So this one would be the dihybrid cross, and this one is just showing you that the only reason there's so many letters is that the capital Y's and the capital R's here are just the two dominant traits. So if that would be a yellow flower and a round pea pod seed, like you see here, then that's the dominant trait. So that's why there's two capital Y's and two capital R's, because there's always two alleles or traits passed on, but you get one trait from each parent. And then that's why when you cross over with the wrinkled green pod over here, the green pod, then you see that now you have dominantly still a yellow and round seed below that, to the arrow below it, but now the letters are different. Now you have capital Y, lowercase y, capital R, lowercase r, and then the next arrow leading down to that is just showing you that now he took the time and crossed all of these. So we're not going to get too crazy about all that, but if we get further along here and people want to do that, we definitely can. That'd be fun to work out with everybody. That'd be really cool. So yeah, once again, let's just go through the terminology here. The P generation is the parents. That's the two things that were crossed to make the first generation of offspring, or the F1 generation. And then if you cross those again, that's the second generation of offspring. And he did this and then came up with the ratios of 9 to 3 to 3 to 1. So you guys can just take a minute to look at that real quick here. Don't have to get too confused about that. Yeah, it's just a representation and a nod to his work. We don't want to be forgetful of the past and people that put in work and took the time to figure stuff out. And he was somebody where his work didn't get credit for at the time, but then people later rediscovered his work and then he got credit. So that's good. And yeah, so if we have questions about this, we can talk about it. DM me on my Instagram, or we can figure something out. We can figure something out. Just comment if you want to do something like that. But yeah, don't be afraid of all the letters and stuff like that. It's not as confusing as it looks. It's just a lot of information, and it's just a lot of different, and it can be different stuff, and it can be overwhelming. So don't let it scare you. Okay, let's move forward. So, as he was getting done with these experiments, he started to make some conclusions about some things, which I'm sure some of you, if you've grown a lot of different grows, or if you've grown a lot of the same cultivar, you probably have some conclusions about how stuff should go as you're doing it, if you have your environment all dialed in. So if something goes wrong or something unexpected happens, you're definitely aware of, okay, well, what's the outlying factor that's causing this. And he kind of was taking really good notes, and he was aware of this too, so he started to make some conclusions here. So with this, we have an idea that he had, and that's widely accepted. It's Mendel's 3 laws of inheritance. So conclusions from his experiments was the genotype and the phenotype stuff that we discussed already, and I want to describe that stuff first because it kind of makes it made more sense to me to try to explain that first, because that's just it's talking about the characteristics of the physical plant that you see. So if you're looking at your plants right now and you see a purple leaf or you see a green leaf, that's a phenotype. But the reason that gene is getting expressed, or the reason you're seeing that being expressed through the plant, that's like the gene, the plant, the seed that it's the seed that the gene is expressing through. If that makes sense. I don't know if I got my stuff jumbled up right there, but that's the gist of it. So genotype is just the genes that are passed on and carried and expressed, and then once you see those in the plant, that's the physical phenotype. That's the physical attribute. So the genes are transferred through the parents to the offspring, and pairs of alleles, which is with the traits. And then during gametogenesis, chromosomes are split in half, and there's a 50% chance of one trait from each parent to come together. And then if they have the same alleles or the same traits being expressed, so like if we go back to the picture, let's go back. So let's do this one. It's easier to understand, easier to see. So if you see the two A's, so if you have two A's and two A's, and that trait is both being passed on, then you would call that, because they're the same, they're homozygous. If the alleles are not the same, like how you would see in figure B, where it's capital A and lowercase a, then you would call that heterozygous. So, as he was making these conclusions, he developed the law of dominance, the law of segregation, and the law of independent assortment. And he started, like we discussed already, seeing in the first generation of offspring a dominant trait being expressed. And like we said, there is still the recessive trait, like you would see. And hybrid just means there's two different parents. So the dominant trait's being expressed in the first generation, but that other trait is still being carried on. It's just it wasn't expressed, so that's why you don't see it coming through the physical attributes. So that's the recessive one, and that is the law of dominance. So from the reference here, this is Mendel's first law of inheritance, and according to the law of dominance, the hybrid offspring will only inherit the dominant trait in the phenotype. The alleles that are suppressed are called the recessive traits, while the alleles that determine the trait are known as dominant traits. So that's just what we described there. I just wanted to give you the reference, what they said about it, so you had the full quote. And then the second one here is the law of segregation, which the law of segregation states that during the production of the gametes, which is the two alleles, so there's two copies of each hereditary factor that segregate, so that the offspring acquire one factor from each parent, or like we said, you get one allele, one trait from each parent. So that's what this is saying. And then when those combine together, they combine, or well, they separate randomly, and then they recombine randomly as well. And when they reunite, and this is called fertilization. And then the part where these combine randomly is also leading into Mendel's last law of inheritance, which is the law of independent assortment. So basically, the law of segregation just means that you have the two traits from each parent, or the two alleles, if it's a dihybrid cross. Though those are two potential possibilities to be expressed. The dominant trait will be expressed in the first generation, just like we've seen with the yellow flower and the round seed pod. And then the next generation of that, there was a different one. There was the green wrinkled seeds. So that's what the law of segregation is talking about. It's just that there's two traits from each parent. You get one trait from each. When they combine, that's fertilization, and this occurs randomly. And then that's also what we call the law of inheritance, or the law of independent assortment, and that's the final law of inheritance. So we talked about this. You guys can read this for a second here. Hopefully everything's going smoothly. I got my notes pulled up on my phone, so I can't really look at the live chat right now and give the presentation. So I'll double back here at the end and look at the live chat. So from the reference, let's just check out what the law of independent assortment is. So this is also this also states that the pair traits segregate independently of the other pair during formation, and as they're separating and then recombining, it's a 50-50% chance, just like you would flip a coin, whether it's heads or tails, like a quarter. If it's heads or tails, that's a 50-50 chance. So that's kind of the same idea. And then like we talked about already, that's why if you have siblings that look similar but not exactly the same, it's because it's from the same gene pool, and it just is a different combination of the two traits, and then that's why you have two different outcomes. So if you have a plant that's short and purple, like the apes in space that I had, and then you cross it to a plant that is tall and green, and then you have the first generation of offspring. If I don't know what the dominant allele or trait is, if I start taking notes and keep popping the same stuff and popping the same seeds, I'll eventually see what's dominant and what's outliers and what's, recessive. And then if you wanted to keep breeding, you would keep crossing those together. But that's further in the future here. We're just trying to build, the first little introduction to this stuff. I hope it doesn't seem like too much. It's just I was trying to do a decent, really good job at explaining this stuff and not putting too much images or words on the screen. So hopefully it's not too much. But yeah, so that's the last law of inheritance. That's the law of independent assortment, and this is why you have multiple seeds. Then each of them turn out differently. It's just different combination of the traits that are being expressed. So that's what that is. So now the next question would be, why is this important to someone that is a breeder? And why was this important to him? Well, because he had a goal and a purpose of what he was trying to do. So the next part of this is purely subjective. So why you breed something, why I breed something, and why somebody in the agriculture industry breeds something is dependent on what you want your goals to be. Somebody that breeds corn is going to have a little bit of a different objective than somebody that breeds cannabis. Maybe not always, but I would say maybe a little bit. But it's still the same principles. So it's the fact that, there needs to be open conversation, because someone that wants to grow vigorous corn and someone that wants to grow vigorous cannabis might be able to work together and utilize modern agriculture to achieve that goal. So being open and willing to work with others will allow a breeder and allow everybody in the home grower community to be able to have faster advancements in the genetics. Because if you watched the live stream last time for the second subscriber event, I believe Culturegen was just saying, sometimes it's a little bit difficult trusting some breeders, and it's just that, and then that also falls on the consumer or us to be due diligent about who we get our seeds up from, because he was saying that it takes a long time to make a proper inbred line and to test them and to go through and do everything you need to do to make a good solid seed line. And he just said it's good to be aware of stuff like that. So I think that's a fair point to mention, and that's why talking about stuff like this is good, because maybe you'll learn something, and if you don't, then we can talk and maybe we'll converse and learn different perspectives, and then we'll learn something. But the reason that you may want to do something like I said, and the reason I want to do something are two different things. So the reason it correlates to what I'm doing with the BBP, Beginners Bridges Project, is because I want to use genetic and selective breeding to stabilize the three crosses that I have made. Two of them accidentally and one intentionally, but I'm by no means a breeder. It's just background information I like to give everybody. If you've watched the channel, then you already know, but I became more interested in learning about this breeding stuff because of the two accidental crosses, which were the runt mints and the JB runts. And I didn't know what to do with the seeds, so I just kept them. And then that turned into me wanting to grow one of them, so I did. And then that ended up being one time when I was growing with Chill Mints OG, or Chill Out OG F3s, and one was a male. And then I collected pollen from it, and then pollinated runt mints. And then, yeah, that kind of led into the beginnings of this. I just kind of kept a little catalog of little videos on my phone and stuff, and just was keeping track of what was going on and everything. And then I just collected the seeds once it was ready to harvest them, and then I just really thought it was a fun thing to do, and I liked it, and I wanted to know what I could do to do it effectively and actually, like I said, stabilize my crosses. But let's talk about those real quick. So there's the runt mints, which is a CRV mints male crossed to white runts, and then there's the JB runts, which is John Boy runts, and that's the CRV mints male crossed to Sensei Seeds critical runts, and those are the two accidentals. And then there is the Chill Mints OG, which is a Chill Out OG male and a Runt Mints female. So what I want to do with this stuff and how I want to make these stabilized genetic is that I would like to take the first parent generation. So that would be if I take 10 seeds of runt mints and I pop them all, and I find the best ones, and then I cross those to one another. The offspring of that would then be the first or the F1 generation, and then if I cross that again, that'd be the F2 generation. And then this ties into the stuff we were talking about, because of then I can see what's dominant and what's recessive, because then I took two of the same, crossed them to each other, and then I took the offspring of that and crossed them to each other. And if there's anything that we don't like hermaphrodites, that was a recessive trait that was expressed, we can try to cull that out if I want to continue. Or if there's stuff where it's like bad larvae buds, or bad terrible lacking smell or something. So you just keep going through and selecting and finding stuff that you like and want, and that's what I'm going to do with all of them. But the reason, another reason you'd wanted to do that is that if you only had a limited amount of seeds and you wanted to make more of the same, you could take those seeds that you have and cross them to each other, and then that will create more seeds, and then also give you more of a genetically diverse seed selection if you do an open pollination. But there's benefits of doing that and not doing that. But the point of this is eventually is to breed out any bad stuff, and then find keeper cuts. And then for my breeders, our beginners breeders project, this is turning into kind of like trying to find some good old or older genetics to work into the currently hybridized crosses I have, and try to use like something more diverse, because the further back you go with some of these certain strains or cultivars, there's different expressions that, or like let's just say terpenes that are not as dominant or not as apparent anymore, because now some of the older crosses or cultivars are gone. You can't get them no more, so then now you don't have access to that gene pool no more, and it's hard to get back to that older stuff. So that's just kind of why I want to start with some older stuff, and skunk number one is something that is talked about quite frequently. So I want to use an already, and another good point is that you always want to use a really good foundation and strong building blocks. So a good way to do that is to use an already inbred line. So I decided to go with Todd McCormick's and Mel Frank's Authentic Genetics Seed Co. Inbred line, skunk number one, and you get 10 regular photoperiod seeds. So you could do a selection from there and try to find keepers and stuff, but I think I'd like to, and I am going through and trying to get more seeds with the intention of finding some more expressions that come out inside the gene pool. So I want to do what I just said. I want to create more seeds, so I'm going to take all 10 seeds and I'm going to try to pop them and cross all the males to the females and collect the seeds from there, and then start going through and selecting what's good and what's not, and then having a keeper cut of that stuff, and then I can start going through, maybe I can start going through one of the rut mints or the JB runs or the chill mints OG after I finish looking through the skunk one, and find one that I like at that, and then cross those two together, and then that's kind of like the intention of it. And this correlates to what we just talked about in the presentation, because once again we're going to be doing crosses of the same exact cultivar, and we're going to do crosses eventually of two of the different ones. So if there's a dominant and recessive trait in the two same ones, what do you think will happen when we start crossing two different ones? So I just think it's a really cool aspect just to think about, and that's just how it correlates with what I'm doing and what's going on and stuff. And really one of the most important things is just to keep popping seeds and to keep growing, the experience it for yourself and take notes and just have fun with it, and yeah, the rest will follow. You if you want to do it, you will. And I believe in you guys, so definitely. I know a couple people out there. Shout out to Jester and a couple other people. They had some stuff going on where they had stuff pollinate in the room and all that, and now they have seeds and everything. But yeah, there's a lot to be done with it. You can breed for whatever you want. If you want it to look really cool, you can breed for that. If you want it to make you feel happy, hopefully you can breed for that. But then you have to do a lot of smoke reports, which that's still fun, but that'd be a lot too. It's all about getting the experience in and recording it, and then wanting to do it. So that's kind of what's going on with that right there. So you can't be afraid to fail, because that's gonna happen, but that's where you can learn to grow just as much as you can from successes. So you gotta take the good with the bad. So when I was buying the seeds from Authentic Genetics Seed Co., I was emailing Todd McCormick and his team or whatever, and just asking them, why or what and how to start with some good genetics. And he was saying, well, or what to breed for in good genetics. And he was telling me that really that the main thing is that I breed and cross something that I like. And he said that the reason he wants to provide the skunk and the Afghani and the Northern Lights and all that stuff is so people can get back to preserving the primary colors in cannabis. That's what it says on his website. But that's neither here nor there. I'm just quoting that because that's what he said and everything. So I'm not sure if everybody agrees with that or not. But that's kind of what I want to do. I want to, and if anybody has any recommendations on some different areas to get some really good authentic skunk seeds, that'd be cool too. I'd like to know that. Shout out to Dab Marino. He gave me one of his skunk selections in his tester bundle that he sent me, and I'm hyped up about that for sure. So at this time, I mean, that's really all that I had planned for tonight. Keeping it short and sweet, trying to at least here. And yeah, I mean, that's what's pop locking and dropping. I got some updates coming to y'all from the seedling tent. Got some updates coming to the harvest and the flowers that we got. Smoke reports coming inbound. We got. I really want to go live tonight and do at least a chop down or two of the Georgia pies, because I saved those ones to chop because, like I said, I wanted to let them go another night. So I'd really like to get back into coming back in a few hours here and getting these chopped, maybe on a live stream, a mobile live stream real quick, and chatting with y'all, hanging out and stuff. So yeah, you guys, like I said, I wasn't in the live chat. Let me go back here. I'll double back and look in the live chat. So I hope you guys have a great day and a great night. I'm glad you guys came by here. All information provided was referenced here, and all diagrams were referenced. I'm really excited. I'm excited to keep getting this stuff going, and like I said, I'm re-vegging a Milktopia, the one that I like the most, and then I want to re-veg one of the Georgia pies I like the most too. And I'm thinking there's this one that's real bright green and has like a really taller structure than the rest. And I just like the nug formation on it, so we're gonna see here. Not sure yet. We'll find out. But if you guys want, let's do another hit here. We'll do another dab hit, another J, whatever you guys want to do. Let's do that real quick, and I won't take up no more of your time. I appreciate y'all. Let's go back to shout out Joe. I just like this. It's really nice. Shout out Joe. Joe. But yeah, we're going to get this pop locking and dropping here. We'll get this final hit going, guys, and we'll skedaddle. It's about to be a fatty. Bam. Alright, here we go. We get this warmed up here. Get this pop locking and dropping, and then I'm outie. Very excited to be getting some Cultigen Seed Co. genetics going. Very excited about that. Shout out to him. Super excited to get P&W cultivar stuff going, which is already going. Shout out to him. Got the Passion Punch back cross to Fruit Punch, and then got his Fruit Pump cross to Caribbean Breeze and Fruit Pump cross to Pink Glizzy. So really excited about some of those. And yeah, just gonna see where it goes here. Some of them we're gonna be getting probably rid of and all that, just because we can't be having too much space be taken up. You know how that goes. It's always a space issue. I got animals too, so I gotta watch out for this freaking cat and dog hair. Cheers, y'all. Oh yeah, that was a good one, guys. All right, well, I hope you guys have a great night here. I think I'm about to wrap it up. I appreciate everybody. I appreciate you guys all being here, and I hope you have a great night, good rest of your week. And like I said, coming inbound, we got some harvest updates. I will let you guys know if I'm going to go live here later tonight and chop down the rest of these here. And then we're going to have update on the seedling tent, and then probably maybe a transplant. Potentially a transplant video. I haven't decided on that yet. Because my goal is to reuse my soil. So I'm going to have to re-amend it with some worm castings. And some other stuff. So we'll get that all taken care of. I'll get that worked out. But yeah, that's what's going on here. Yeah, we're going to get out of here, guys. I'm glad you guys came by. Have a good one. Be safe. Stay warm. If it's cold where you're at, if there's snow, be careful. Peace and love to y'all. Many blessings. One love. I'm out. Special thanks to everybody once again. Have a good one, everybody. Peace.